Welcome to the introduction to veterinary endocrinology. Our first topic under this section is the endocrine system. In this presentation, students must be able to describe the basic concepts and nature of hormones, identify the different endocrine glands, compare endocrine from exocrine glands, and to classify hormones according to different parameters. So there are different parameters that we can use in order to identify and classify hormones. Example of this are its chemical structure or chemical nature, as well as its site of production. So the hormones later on can also be classified according to its main function, whether it is metabolic in nature or it is mainly involved in reproduction. So we basically define endocrinology as a study of the structure and the physiology of the endocrine system, particularly the endocrine glands, and their secretory products, which are the hormones. So the basic function of the endocrine glands and the entire endocrine system is to produce hormones. When we say hormones, these are specific messengers that regulate important body functions. So it is said that the endocrine system and the nervous system work together and complement each other, but they tend to work at different speeds. The nerve, which is a component of the nervous system, respond within split seconds, but their action soon fades. Meanwhile, the endocrine system, particularly the hormones, have a longer lasting effects and act over hours, weeks, and years. So comparing the adjustments to stimuli between the endocrine system and the nervous system. So the endocrine system generally has a slower adjustment in response to stimuli than the nervous system. So for example, some noxious stimuli, such as a hot burner, require an immediate response if injury is to be avoided. So the exposure to a noxious stimuli, such as hot burner, will tend to activate the nervous system so that there is a rapid reflexive response that will avoid that particular noxious stimuli. When we are going to compare that with the responses from the endocrine system, so again, the responses from the endocrine system is said to, it can be measured for a longer time, in hours, months, or years. So, for example, the events in the development of puberty will take time, and this is said to be, of course, hormonally dependent. So the integration of the nervous system and the endocrine system during response to a stimuli is referred to as the activation of the neuroendocrine system. So the classic example of the neuroendocrine integration in farm animals is the milk letdown reflex in cows, which has a 6 to 8 minutes delay. So there is generally a 6 to 8 minute delay between the time of initial udder stimulation and full milk release. So when we say initial udder stimulation, that refers to the presence of the stimuli, and this stimuli includes suckling and manual stimulation. So there is a 6 to 8 minutes delay because the milk letdown, even if the milk is already present in the udder, the milk letdown is still dependent on a specific type of a substance, and that is the hormone oxytocin. Oxytocin is, of course, produced from the hypothalamus, and it is secreted in the posterior pituitary gland, which is located in the head. So the release of the oxytocin will depend in turn on the neural impulses from the sensory neurons in the udder. So when there is a specific stimuli, such as suckling or manual stimulation, it will send a sensory signal or a neural impulse from the sensory neurons in the udder, so that when the udder is stimulated, the neural signals will travel along the afferent nerves, of course, to the CNS, to the spinal cord, and into the brain, where they will be relayed to the neurons of the posterior pituitary gland. So the neurons of the posterior pituitary gland contains the hormone oxytocin, which is secreted from that particular gland or from that particular endocrine gland. So upon secretion from the posterior pituitary gland, what will happen is the blood will the hormone will be secreted in the blood, and it will travel now via the venous system to the heart, and from the heart the blood will travel to the lungs now for oxygenation. Then again it will travel back to the heart, to the arterial system. So from the arterial system, it will travel to the different parts of the body until it will reach the udder. And that is the time where the milk letdown will happen. So hormones are secreted by endocrine tissues, which are carried straight into the bloodstream to other cells of the body. So they help regulate metabolism, digestion, reproduction, electrolyte and fluid balance, growth, as well as the development. So hormones are again secreted from their source, and that is the endocrine gland, directly into the bloodstream. Then the blood will carry the hormone to the target cells which respond to it. They are produced in very small quantities, so in other references, they are said to be produced in picograms and are biologically very active. So an example of this is the hormone adrenaline or epinephrine. It is active even at a concentration of 1 in 300 million parts. So the excess and the deficiency of hormone will cause serious disorders. The extra hormones are not stored in the body and are excreted out. So the endocrine glands are ductless glands, and it releases hormones into the bloodstream. Its action could either be paracrine or endocrine in nature. When we say paracrine action, the hormones act on the neighboring cells. And this is important in the digestive system. So the gastrointestinal tract is considered to be the largest endocrine organ in the body, and the endocrine cells within it are referred to collectively as the enteric endocrine system. There are three of the best studied enteric hormones include gastrin, cholecystokinin or the CCK, and the secretin. So gastrin is secreted from the stomach and plays an important role in the control of gastric acid secretion. Cholecystokinin is a small intestinal hormone stimulating secretion of the pancreatic enzyme as well as the bile, and the secretin is another hormone secreted from the small intestinal epithelial cells stimulating the secretion of bicarbonate, rich fluid from the pancreas and the liver. So in contrast to the endocrine glands like the anterior pituitary gland, in which essentially all cells produce hormones, the enteric endocrine system is diffuse, meaning single hormone secreting cells are scattered among other types of epithelial cells in the mucosa of the stomach and the small intestine. For example, most of the epithelial cells in the stomach are dedicated to secreting mucus, hydrochloric acid, or pepsinogen into the lumen of the stomach. So scattered among the secretory epithelial cells are the G cells, which are endocrine cells that synthesize and secrete the hormone gastrin. Being a hormone, gastrin is secreted into the blood, not into the lumen of the stomach. Similarly, other hormones produced by the enteric endocrine systems are synthesized and secreted by cells within the epithelium of the small intestine. So those hormones, the enteric hormones, act via paracrine action, meaning that they act on neighboring cells. Another is the autocrine action. These are the hormones that may be generated and act on the same cell. This diagram shows how the hormones are released into the bloodstream and the blood carrying the hormones to target organs. So we have here the endocrine cells, and they secrete the hormone directly into the bloodstream. So these small triangles here are the hormones. They travel into the blood and they reach the target cells with the hormone receptors. So this diagram shows the structure of a typical blood vessel. We have here the presence of the endothelial cells that line the blood vessel. We also have here the presence of the vascular smooth muscles. So the vascular smooth muscles, as you can see here, are present on the arteries as well as on the venules, particularly the larger venules. As you can see here, the capillaries do not contain the vascular smooth muscle, so that they are considered to be the major site for the exchange of nutrients, hormones, gases, and the macromolecules. So from the bloodstream, so the hormones can be exchanged, or it can pass through the blood vessel, particularly the capillaries, to reach its target tissue containing the receptor for that particular hormone. This diagram shows the two general types of glands in the body. So we have the exocrine gland and the endocrine gland. So when we say exocrine glands, these are glands that secrete substances. Example of these are saliva and milk, which are carried through ducts to a specific site in the body. Of course, the endocrine glands are ductless, and of course, their secretion, which are the hormones, are directly secreted into the bloodstream. So this diagram shows the exocrine gland. So the exocrine gland contains the ducts that conveys its chemical secretion to the outside. For the endocrine gland, so we have here the presence of the blood, the capillaries, whereby the exchange of the hormones will occurs. And in here, we have here the presence of the endocrine cells. These cells will secrete the hormones directly into the bloodstream. So let's now proceed to the signaling molecules. And the signaling molecules are important for the function of for the hormones to be able to produce its function. So the ability of the cells to communicate through the chemical signals is necessary, and the cells can receive a message, transfer the information across the plasma membrane, and then produce changes within the cell in response to the message. Cellular communication has developed as a means to communicate with the environment, produce biological changes, and if necessary, ensure survival. So there are different forms of signaling. So the first type of signaling mechanism is the endocrine signaling. So in this type of signaling, so the signals from the distant cells that originate from the endocrine cells usually produces a slow response but having a long-lasting effect. We also have the autocrine signaling. So this is produced by signaling cells that can also bind to the ligand that is released. The signaling cell and the target cell can be the same or a similar cell. So this comes now from the prefix auto, meaning self. Another type of signaling is the paracrine. This is a form of cell signaling in which the target cell is near the signal releasing cell. Next, we have the direct signaling. So this occurs by transferring signaling molecules across gap junctions between neighboring cells. So these are the different forms of chemical signaling. We have the autocrine. So in autocrine, the cell targets itself. So it produces a signaling molecule, then that molecule will target the cell itself. We also have the signaling across gap junctions, and the cell targets a cell across the gap junctions. So we have here the signaling cell and the target cell, and we have the presence of the gap junctions. We also have the paracrine. Again, para comes from the word near. A cell targets a nearby cell. So we have here the signaling cell and the target cell. For when we say endocrine, a cell targets a distant cell through the bloodstream. So essentially, when we say endocrine, the signaling cell is distant to the target cell, and the signaling molecule will need to travel via the blood to reach its target cell. So this diagram shows an example of the paracrine and the autocrine signaling in the heart. So in this diagram, so we have here the presence of the endothelial cells. The endothelial cells secrete the signaling molecules or signaling proteins in blue dots. And these signaling proteins will target receptors that are found on the cardiomyocyte, the inflammatory cell, and the fibroblast. So in the autocrine signaling, so the endothelial cell in this example is shown, and the endothelial cells produce the signaling molecule, and the signaling molecule will bind to the receptors that are found on the same type of cell, which is the endothelial cell. So for a hormone to produce its response, it needs to bind first to its receptor. So the receptors are protein molecules in the target cell or on its surface that bind ligands. So the ligands that we are referring to here are the hormones. There are two types of receptors, the internal receptors and the cell surface receptors. So the first type of receptor are the internal receptors. So from the name itself, they are internal. So they are found in the cytoplasm of the cell, and they respond to hydrophobic ligand molecules. So when we say hydrophobic ligand molecules, these are water-fearing, or these are lipid-soluble molecules or lipid-soluble hormones. So when the hormone will enter the cell, so it will bind to the receptors in the cytoplasm. Then after binding, there will be a conformational change, exposing the DNA binding site on the protein. The ligand receptor complex moves into the nucleus. It will bind to the specific regulatory regions of the DNA, and it will promote transcription. So once inside the cell, many of these molecules will bind to proteins that act as regulators of mRNA synthesis to mediate gene expression. So we have here an example of the mechanism whereby the intracellular receptors will bind its ligand, which are the hydrophobic signaling molecules. So again, when we say hydrophobic, these are lipid-soluble. So hydrophobic signaling molecules, since they are lipid-soluble, they can diffuse readily across the plasma membrane. So this is the plasma membrane of our target cell. We also have here the cytoplasm. We have here the nuclear membrane, the nucleus, and the DNA. So the hydrophobic signaling molecule will readily diffuse in the cytoplasm, and it will bind to the receptors that are present in the cytoplasm. After binding, it will create a conformational change, exposing the DNA ligand binding site. After that, it will interact with the DNA in the nuclear DNA to promote transcription, thereby regulating the gene expression. Another type of receptor are the cell surface receptors. So these are membrane anchored or integral proteins that bind to external ligand molecules. They perform signal transduction, converting an extracellular signal into an intracellular signal. They are also called as cell-specific proteins or markers because they are specific to individual cell types. So for the cell surface receptors, we have three domains. The first domain is the extracellular domain, and this is the domain that is located outside the cell surface. Its main function is, of course, since it is located on the outside, it will be the one to bind to the first messenger, which is the hormone. And this is considered to be hydrophilic in nature. The second domain is, of course, the transmembrane domain. Of course, this is the one that is located within the cell membrane. And on the inside of the cell, we have the intracellular domain. This is also known as the cytoplasmic domain. And it is the one that will interact with the molecules, leading to the generation of the second messengers. This diagram shows a typical structure of the cell surface receptors, showing the three domains. The first one here is the extracellular domain. The second part here is the transmembrane domain, and the third part is the intracellular or the cytoplasmic domain. So there are different types of cell surface receptors. So some examples are the ion channel link receptor. We also have the G protein link receptor and the enzyme link receptor. So what is this ion channel link receptor? So this type of receptor will bind a ligand and open a channel through the membrane that allows specific ions to pass through. So that is why it is known as the ion channel link receptor. So when a ligand binds to the extracellular region of the channel, there is a conformational change in the protein structure that allows the ions such as sodium, calcium, magnesium, and hydrogen to pass through. So this type of receptor are widely used within the nervous system to allow neurotransmitters to stimulate cells. So this is an example of an ion channel receptor, particularly a gated ion channel. So in this particular diagram, we have here the presence of the cell membrane, and we have here the presence of the ion channel receptor. We also have here the signaling molecule and the ions. So when the signaling molecule bind to the ligand binding site of the ion channel, so it will activate the channel, opening it and allowing the ions to pass through the cell. Another type of cell surface receptor is the G-protein coupled receptor, or the GPCR. The GPCRs are considered to be the a large family of cell surface receptors responding to a variety of external signals. Binding of a signaling molecule to a GPCR results in a G-protein activation, which in turn triggers the production of any number of a second messengers. So that is why it is called as a G-protein coupled receptor, because it involves the G-protein. This is a typical diagram of the G-protein coupled receptor, showing here the presence of the G-protein. Another type of cell surface receptor are the enzyme link receptor, particularly the receptor tyrosine kinases. So these are membrane receptors that attach phosphate to tyrosine. A receptor tyrosine kinase can trigger multiple signal transduction pathways at once. So let's now proceed to the different examples of the endocrine glands. So examples of the endocrine glands are the hypothalamus, also have the pituitary gland, the pineal gland, the thyroid, adrenal gland, ovary, uterus, placenta, testes, as well as the pancreas. According to function, there are those glands that are considered to be exclusively endocrine. So for example of this is the pituitary gland, thyroid, parathyroid, thymus, and the adrenals. Meanwhile, there are also those glands that are said to be partially endocrine. Example of these are the pancreas, gastric, and the duodenal epithelium, the gonads, and the placenta in females. So the pituitary gland is considered to be exclusively endocrine because its main function is to produce hormones, the hormones, the trophic hormones. We also have the thyroid gland. Of course, the thyroid is mainly responsible for the production of the metabolic hormones T3 and T4. For the parathyroid gland, of course, its main function is to produce the hormones, yung parathyroid hormone, as well as the calcitonin. The same is true for the thymus and the adrenals. The pancreas is considered to be a partially endocrine gland, because in the first place, the pancreas has two parts. It is not mainly an endocrine gland, but it also has an exocrine part, the exocrine pancreas, which will produce the pancreatic enzymes or the pancreatic juices. The same is true for the gastric and the duodenal epithelium. So aside from producing the endocrine hormones, these parts of the digestive system will also produce gastric juices and mucus. The gonads, for example, in males, will produce also the gametes, the spermatozoa, aside from producing testosterone. The same is true for the ovary. It will produce the ova, aside from producing the hormones, estrogen. So hormones can also be used to regulate body functions. These body functions include growth, lactation, birth control, estrous cycle, and others. So for example, the anabolic steroids can be used to promote growth. The growth hormone or the somatotropin can be used to regulate lactation. The estradiol and the progesterone are also important as a birth control. The PGF2 alpha is of course important in the regulation of the estrous cycle. The follicle stimulating hormone and the equine chorionic gonadotropin are important for its function in superovulation and in bryotransplant, as well as, of course, we have the oxytocin, which is very important for the induction of parturition. According to the type of action, so there are those hormones that have a primary function for reproduction. We also have those hormones that are considered to be not metabolic in nature. So example of the hormones that are metabolic in nature, for example, we have the thyroid hormones. They regulate metabolism. We also have those hormones that are primarily for reproduction. Example of these are the typical hormones estrogen and the testosterone. According to its chemical structure, so the hormones can be classified according to its chemical nature. We have those hormones that are made up of proteins and peptides. We also have the steroid hormones, the hormones that are made up of fatty acid, as well as the modified amino acid. The releasing hormones are considered to be proteins, or they are made up of proteins. And an example of a releasing hormone is the gonadotropin releasing hormone. For the steroids, so example of steroids are the estradiol, the testosterone, and the progesterone. For the fatty acids, so we have the prostaglandin, PGF2 alpha, which is made up of fatty acids. And of course, we have the modified amino acid, thyroid hormone, as well as the catecholamines, epinephrine, and the norepinephrine. So peptide hormones are made up of small chains of amino acids. In other words, they are considered to be small proteins. So too large or too water soluble to enter the cells. So their property is that they are water soluble and they diffuse through the tissue readily and circulate freely in the bloodstream. Examples of the peptide hormones are releasing hormones. From the example is the gonadotropin releasing hormone from the hypothalamus. We also have the hypophyseal hormones. Example of this are the luteinizing hormone and the follicle stimulating hormone from the pituitary gland. We also have the neurohypophyseal hormones from the posterior pituitary. We have the vasopressin, the ADH, and the oxytocin, and the gonadal activin and the inhibin. The gonadal activins, so the activins are important now for the stimulation of the FSH, and they are produced in various endocrine tissues, and the inhibin has an opposite effect. So when we say steroid hormones, they are mainly derived from cholesterol, and they are synthesized in the adrenal cortex, the sex glands, and the placenta. They are considered to be lipid soluble, and they are not, again, very water soluble, so that they require transport proteins to carry them throughout the body via the bloodstream. According to the site of action, so we have here those hormones that act on the cell surface receptors. So they are usually the hydrophilic ones, no, or the water soluble hormones. Example are the peptide hormones, the eicosanoids, the PGF2 alpha, and the catecholamines. We also have those hormones that will act on the receptors located on the cell nucleus or the cytoplasm. So these are the hydrophobic ones. These are the lipid soluble hormones. Example are the steroid hormones, as well as the thyroid hormones. So again, the protein and the peptide hormones, the catecholamines and the eicosanoids, they find their target receptors on the plasma membrane of the target cells. They are considered to be hydrophilic in nature, meaning that they are water-loving, and they cannot cross the cell membrane. Because they cannot enter the cell readily, they need a second messenger to carry out effects inside the cell. And this mechanism, whereby the hormone utilizes a second messenger in order to carry out its effect, is known as the signal transduction pathway. So this diagram shows the specific examples of the cell surface receptors. So as you can see here, we have here the plasma membrane, the extracellular, and the cytoplasm. So as you can see here, the cell surface receptors mainly has three domains. Now we have the extracellular domain, we have the transmembrane domain, and the intracellular domain. So the intracellular domain is the one that is found in the cytoplasm. The extracellular domain is the one that will bind to the ligand or to the hormone. So this is an example of the beta-adrenergic receptor. So this will be the one that will, its ligand are the catecholamines. For the insulin receptor, of course, this will bind insulin, which is an example of a peptide hormone. You also have the epidermal growth factor receptor.